Hey everyone, it's Ms. Hardy. I'm recording for Biomedical Technology on Tuesday, September 29th. We are officially closing the chapter on genetically modified organisms and transgenes and transgenic organisms, and we are starting cloning. All right, so what you're going to see me do is share my screen with you. We're going to click right into that homepage of Biomedical Technology, clicking on the agenda. We're going to see that for today, we will have our discussion post, which has already been posted for you, and making sure that we're doing those every day. You never know when I take them for a grade. So I took one last week, and it would just happen to be a day that not many of you had done it. So keep in mind, those have to be done every day. For today, after the discussion, we're going to do the cloning lecture, which is going to be notes. I have gifted you the note guide that I made. Okay, it looks like this. There's already a picture posted of it. Okay, what I've done is taken that extra long PowerPoint and I've condensed it to the pieces that you need and the pieces that I'm going to go over today. Okay, so I'll be skipping around through the slides. And again, I like to call this picking out the meat or picking out the important stuff of the material that we're covering. So this is what you're going to need to know for your test. Okay, one thing I do want to talk about as we're going through my note guide, guys, like right here, I know it's a mirror image to you, but you'll be able to pull it up on your computer, is that I want to talk about how to take good notes, because your job yesterday was to take cloning notes on your own. And so I wanted to see kind of where you guys were in terms of taking notes, and we're still kind of all over the place. We're either writing too much, we're not writing enough, we're just copying and pasting. So I wanted to make sure you guys understand that I do think the best notes are handwritten, because this makes you accountable for the information. You're not just copy and pasting or mindlessly typing, okay? And I do think if you're going to type them, then make sure you use colors, okay? So I use colors to fill this out. For me, I feel more confident giving you this lecture because I did these notes this morning. Even though I've given this lecture 10 times before, this helped me remember the things that I wanted to talk about. Okay, so we're taking good notes again. Heading back to, obviously our discussion is here, it's on stem cells. We're going to click on modules, and we're going to keep all of this under Unit 3, okay? Under Unit 3, the challenges to biomedical research. Excuse me, I have skipped through. Here we go. All right, Unit 3, we're going to keep everything together because Unit 3 is all about the challenges. So transgenic organisms, genetically modified organisms, were just one of the challenges, okay, to biomedical research. This is a second challenge, okay? So clicking into this PowerPoint. Hold on one second. Technical difficulties, guys. I may have to do a preview of this PowerPoint. Here we go. Okay. All right. So what is a clone? And I've started that here on the top of my document. It's a precise copy. Precise meaning down to the very last genetic detail. It is a very precise copy. Okay. So have you ever heard of Dolly? Dolly was the first mammal we successfully cloned and carried to term. Okay, so when we throw around the word cloning, we know clone means a precise copy, but we're going to talk about two separate types of clones. Okay, Dolly the sheep was considered a reproductive clone. Okay, so we know that there are two types, therapeutic and reproductive. Dolly was a reproductive clone because we made a brand new organism, and that is detailed in your notes. Okay, so that's the definition of a reproductive clone, is we made a brand new organism, whether that be an animal or a human. Okay, she was born in 1996, and she died via euthanasia, which is putting her to sleep, in February of 2003. As you can see here, we're going to clarify this a little bit more, but basically we took cells from a pregnant sheep and introduced them to the uterus of our surrogate sheep. And we know surrogate is not the original mother, okay? And Dolly was born. Dolly had the DNA of her source, the original. She was born, she had the DNA of the original sheep, meaning she was a precise genetic copy of that original mother sheep, okay? She was put to sleep in 2003 after doctors were able to detect a progressive and rapid growing lung disease, and that you'll find is one of the common issues with clones, is that they do have a lot of health issues. Okay, even though she'd only reach half of the typical lifespan of a sheep, that's one of the things we're noticing with these clones, is that they have more health problems. Okay, so SCNT, you need to know this. This is somatic cell nuclear transfer. Somatic just meaning living cell of the body. Okay, so there's a lot of information listed on this site. There are basically 5 steps to SCNT. Here's where I'm going to pull out my notes, and I'm going to give you what my notes tell me. Okay, so we're going to start with an unfertilized egg from a female. Okay, so we know that eggs come from females, sperm come from males. So we're going to start with an unfertilized, meaning it's just an egg. There's no sperm. They haven't met. They haven't created a baby yet. It's just the egg from a female. We refer to that as an oocyte. Okay, oocyte, because ovary is oo. Okay, so an oocyte is an unfertilized eggs, and eggs live in ovaries. Okay, what we do, I'm going to admit some people into the room. Okay, hi, try to pick up and follow along where we are. So the 5 steps of SCNT, okay? So we have our oocyte. It's our unfertilized egg, okay? What we're going to do is we're going to take this oocyte and we're going to remove the nucleus. And we know that within the nucleus, most of us who have already had biology know that within the nucleus is our genetic material. It's what makes the cell special. It's what makes the cell what it is, okay? So we're going to actually take out that nucleus of the oocyte. Then we're going to have our donor cell. Okay, our donor cell has its own nucleus. We're going to take that nucleus and put it into the original oocyte. And what happens is that whole cell reprograms itself based on the new genetic material it received from that new nucleus. So a couple of different terms here. We have oocyte, which means unfertilized egg. And then the process of removing the nucleus from that original oocyte is called enucleation. Enucleation, okay, and you're going to see that on my note guide. All right, once we have inserted that new information from our donor cell, we are now creating our clone. Okay, the new nucleus is going to reprogram that egg cell, that oocyte, and then what we're going to do is take this whole new thing that we've made and we are going to put it into the surrogate to carry to term. That is creating the clone. Okay, so what we're doing is we're cloning wherever we got that donor egg from. Just like with Dolly, okay? Dolly's mother, who birthed her, is different from the clone of which she was made, okay? So a couple of different terms you need to know. Oocyte, enucleation. You need to understand the term surrogate, okay? All of those things. So what I've done is I've taken the meat out of these slides that you see here, out of all of these words, and I've given you the step-by-step process here on the note guide that I made for you. So if this is neater than the note guide you made for yourself, you may want to copy mine, okay? And again, it's already posted. I'll show you before the end of the lecture. Moving forward, we do have some challenges to reproductive cloning. So that's what we call this. If you carry the baby to term, carry the fetus, nucleus, embryo, whatever to term, then we call that reproductive cloning, okay? Reproductive cloning. So that's when we create a new organism, all right? So there are some challenges to that, okay? First and foremost, there's a very low success rate. Out of 100 times of us implanting that new cell into a surrogate mother, we only achieve like 3% live births, okay? So that's a really, really, really small amount, 0.3%. Really, really small amount of success in achieving live births, okay? Typically, we have fetal development issues, so it may be where the fetus stops developing or it develops with certain disabilities, physical impairments, things like that. We also notice, just like with Dolly, there are a lot of health problems, even if we're able to carry the fetus, the baby, animal, whatever, to term. And carrying to term means that we are able to birth that child, okay? So we have a lot of issues happening here. One of the biggest things that you need to know, and I know this will most likely be on your test, is that there is a certain syndrome. It's called large offspring syndrome, and that tends to happen when we're working with reproductive clones. The baby or the fetus or the embryo tends to go 20 to 30% larger than it's supposed to be. So think about a human baby, okay? If a human baby at its biggest is supposed to be 10 pounds, Okay, and even that can be pretty hard to push through a birth canal. If it's 20 to 30% more of that, it can be upwards of 15 pounds. Okay, and so that our bodies aren't made. Look at Dolly, look at Sheeps. They're not made to push out something or birth something that's 20 to 30% larger. Okay, so we call that large offspring syndrome, and what that makes is for a very difficult delivery, which again can cause death of our new reproductive clone and or the surrogate parent. So those are our challenges to cloning. So we've kind of toyed around with this already during this lecture, but the differences in therapeutic cloning versus reproductive cloning. So I'm actually going to do reproductive cloning first, okay? So there's not actually much information in here other than this slide here. We know that SCNT, somatic cell nuclear transfer, is basically done in both episodes of cloning. There's only one difference, okay? So with reproductive cloning, we go through the whole process. It's implanted into the surrogate mother and hopefully is carried to term and birthed, and we now have a reproductive living, breathing clone. The difference and the opposite to that would be therapeutic cloning, okay? So therapeutic cloning is, the big difference is it's never implanted back into a surrogate parent. It grows in a lab, and then we go from there, okay? So just making sure we understand what a reproductive clone is. A reproductive clone creates a brand new organism, okay? That's all you really need to know about a reproductive clone. Dolly is considered a reproductive clone. The opposite to that is therapeutic cloning, and that's where we're going to spend a lot of our time talking about today, okay? So therapeutic cloning, there's a lot of information, as you can see up here as I'm scrolling through, all of the information on therapeutic cloning. I shortened it for you, and I'm going to go over my notes. So therapeutic cloning, the biggest difference is instead of implanting into a surrogate mom, we actually let it grow in a lab. And in the lab, it grows in a dish, a culture plate or a petri dish, and we allow it to grow and start to multiply, okay? And it starts to divide. And we let it divide until it reaches that blastocyst stage, okay? So it becomes a blastocyte, okay? Blastocyte, okay? Once it gets to that stage, we open up that blastocyte. So again, we're talking about on the micro level here. Very, very, very difficult work. Super cool, very difficult. We open up the blastocyte and we remove what we call stem cells. And that's what your discussion was on today. Stem cells come in many different forms, and they're really cool, and they're very, 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 very valuable, okay? So we remove the stem cells. Why would we use stem cells? All right, here's where it gets kind of sticky, okay? Stem cells are a perfect match tissue. So if you take stem cells from my body and you begin to work with them, they're a perfect match to my body, okay? Stem cells are also going to match what the neighboring cells do. So if we needed to grow new liver cells, we could take stem cells from this process, put them in the same dish as liver cells, and they would begin to create liver cells. You see where I'm going here. The goal of therapeutic cloning is to treat diseases, okay? I can already tell you 10 different diseases that I'd love to see what stem cells could do for them. Stem cells are packed full of everything we need to start treating diseases. But we also know with that comes a price, okay? So we know that stem cells are important. We know that there are 3 types of stem cells we need to know for this class. There is totipotent, pluripotent, and multipotent, okay? We know that totipotent come from recently fertilized eggs, and they can become any cell within the human body, okay? Pluripotent is embryonic, and embryonic sounds like embryo, which comes from that initial phase of unfertilized egg becoming fertilized. And then we have multipotent, okay, which is an adult stem cell. Multipotent are the adult stem cells that we normally find in the bone marrow, so deep within the inside of a bone, where you'd have to do like a bone marrow biopsy. Difficult to come by, difficult procedure, etc. And those are also the most limited. They only become very specific cells. There's what the body tells them that they need, okay? So totipotent can become any cell, pluripotent can become a good amount of cells, and then multipotent can be specific cells. And for the purpose of this class, just knowing those definitions will help you be successful, okay? So that's what all of this is telling us. This is telling us that once we open the blastocyte, we're going to reach the stem cells and we can take them out, put them in their own culture plate, and they will begin to form clones. Okay, so this again goes over the different types of stem cells. So your totipotent is kind of your umbrella cell, and then you have pluripotent and multipotent underneath that. So pluripotent sounds like plural. It can become many different types of cells, okay? And then you have multipotent, which are your adult stem cells, and those are kind of already wired, so those only become certain stem cells, okay? All right, so again, we've discussed SCNT, somatic cell nuclear transfer, and how that process stays the same no matter if we're doing therapeutic cloning or reproductive cloning. And the goal of therapeutic cloning is stem cell research and use, and the goal of reproductive cloning is to create that new organism, okay? So there's a couple of different pictures here, okay? All right, so again, the potential of stem cells would be that perfect match tissue, neighboring cells, and they can become that type of cell. That's what differentiate means. They kind of take on the role of their neighbor cells. And then again, we hope that we can begin to treat diseases with this process. Okay, so in the corner of my notes here, I've included a debate section. It's in purple. Debate is in purple because there's a lot of debate over stem cells. Okay. And keep in mind, we love to debate in this class, so you may need this information in the future. Okay, so the debate over stem cells is, if we use embryonic stem cells, which again, embryo meaning coming from when a fertilized egg is made, fertilized meaning sperm and egg meet, okay, are we potentially destroying something that could become a living organism? So all of us at one point were embryos, okay? At one point in our mother's womb, we were an embryo. And the debate over using embryos to make or to achieve stem cell research says, are we potentially destroying a living thing? All right, so taking it back to this slide up here, where it says it's creating embryonic stem cells that are genetically compatible with the recipient, okay? So in order to have that, you would have had to fertilize an egg, okay? Fertilize an oocyte with a sperm. So you have a fertilized egg, okay? And then we would take the stem cells out. We would allow it to multiply just like it would if it was in your body and you were pregnant, okay? And we are allowing it to multiply, and then we're stopping that process to pull out those stem cells, okay? The debate over that is, again, are we potentially destroying what could be a living, breathing organism? Are we destroying what could be a human, etc.? Okay, opponents say that once the sperm and egg meet and become an embryo, that we are destroying a living, breathing potential human. And so that is the biggest debate. All right, and so again, this is going to dive deeper into some of those issues that we talk about in this class, such as embryonic stem cell research is the big one. Abortion, we talk about that because again, that's a challenge to biomedical research. And is this a form and all of that? Now, again, opinions aside, we're looking at the science and the facts here. Okay, so that's your debate section. It is heavily monitored by the federal government as far as stem cell research. At one time, they actually stopped more federal money going into the research, and that was back in 2001. People didn't feel that it was right. It felt very against nature to mess with making precise genetic copies of anything. And then actually in 2009, which seems so far away now, 11 years ago, President Obama overturned this and allowed for more stem cell research. Okay. Currently, the federal government doesn't have a hard rule. They don't say no way you can't work on somatic cell nuclear transfers and cloning, but there are a lot of procedures in place to make sure that you're working within your barriers. And so the Food and Drug Administration, the FDA, controls a lot of what we do. and they also control a lot of the permissions that are given to continue your work within somatic cell nuclear transfers. And we know that somatic cell nuclear transfer is how we achieve either a therapeutic clone or a reproductive clone. The only difference being what happens once we change the genetic information. Do we allow that embryo to continue growing within a surrogate mother and produce a reproductive clone, or do we allow it to grow to the blastocyte stage, stop, and pull out what we need, which would be the stem cell research? Okay, public opinions here. So again, 60% of Americans 63% of Americans feel it's morally wrong to clone animals. 88% of Americans feel that it is morally wrong to clone humans. And 59% of Americans believe it is acceptable to use embryonic stem cells, which again would mean stopping the growth of that embryo to a fetal stage and pulling out those stem cells, and they believe that that is morally acceptable. So that's important when we talk about debates in this class and what we think, where the fine line should be drawn with biomedical research. Okay, so this is your PowerPoint. It's a lot of information, and it's difficult when you guys aren't in class for me to have you practice saying things with me and going over the steps, but I hope that I have given you information here in the, I'm going to share the screen again. I hope that I have given you this information in a little bit neater of a note guide. I know it was difficult for you guys to take your notes yesterday. It was information that you just weren't sure how to organize. So I'm going to click on my note guide and just kind of let you look through it really quickly. My advice would be, again, keeping in mind that I've done my colors for a reason. So I did my SC. I'm now seeing you guys can't see my screen. Okay, so if you click here on this cloning notes, it's going to pull up this image right here. Okay, it's going to pull up this image. And it is going to, if you click on that, it's going to pull up this image right here. Okay, so what I did was I listed my SCNT in one color, my challenges to cloning in one color, my therapeutic cloning, my reproductive cloning, and then the debate over it all. And if you continue to have questions or confusions, give me a call and I can answer your specific questions. I've tried to make it as simple as possible. And again, to be successful in this class, a biology background is really, really good. And even an anatomy and physiology, which a lot of you have had health science one before. But if you have any questions, comments, or concerns, I'm here to help you, okay? So your goal today is to take your notes from what you had yesterday and turn it into something that you can really work with, okay? So I'd like to see you have something like this to refer back to versus having something like what I got from most of you yesterday, which was just a lot of paragraphs of listing things. We don't do these notes for copy and paste. We do these notes to learn from. And if you were to be in class, I might say, hey guys, get out your note guide from Unit 3 cloning and use it to answer these questions. And so if it were me, I'd want something like this that was organized but also helpful versus just paragraphs of information, okay? Does anybody have any questions, comments, or concerns? Anybody in Zoom have any questions, comments, or concerns? Okay, so again, today, review this. This is coming back. Okay, this is coming back tomorrow. All right, so I won't tell you how and I won't tell you why, but this will be back. Okay, thank you guys so much.